All right. So for the next part of the lecture and the second part, half of it, and this is the third lecture of DNA sequencing technology. So the first part, we talk about how great this technology is, what kind of biological applications we can have, and how that can help us in terms of personal genome sequencing and how that can help us clinically in the future. In the second lecture, we really go into deeper on the data analysis part. We talk about refined alignment, a couple of other things. So today, the focus will be the variant prioritization. So after identity variation, where we're going next. So just to recap a little bit from last lecture. So we talked about the mappability issue initially, that we know that human genome is really repetitive. This figure is not very clear, but you can see that after class. And one of the colors, which is blue here, is the human genome, and the X here is the length of the read. So if you go to 20 nucleotide length, and it's about 75% of human genome you can uniquely map to. If you go to 30 base pair of the reads, it goes to 78%, and it goes to this plateau. Even though you increase the length of the read, it will not increase your mappability, right? And because of the repetitiveness of the human genome. And the second point of this is a non-unique mapping can cause analysis error, especially when we identify genetic variants. Okay, sometimes we know that there's a repetitive part of the genome. You find one part, and the second part is so similar to each other. Once you identify the variants, you think you get variants, but it's very possible that it's just because there's another similar pattern in the genome as well. And the second part is for the cumulative measurements, such as RNA-seq and copy number variations. So if you see the number of reads in one region is different from the other one, that is potentially caused by the mappability issue as well. And the third application of mappability is when you design the capture assays, you really don't want to target on the repetitive regions. So you want to do this type of mappability analysis before you design the capture assays. And the last point here is the mappability is region-specific and alignment algorithm-specific. So if you focus on the exon sequencing or you focus on one part of the genome you prefer to work on in the future, and you want to do the mappability analysis within that specific area and using the algorithm that you will be using in the future. Because different algorithms have different sensitivities and specificities. So they have different tolerance on whether there's a gap and things like that. So mappability will be different. Okay? Any question about this part? All right. And we further talked about refined alignment. So in the second lecture of the whole class, we talk about, we spend an hour also talk about alignment algorithm. But just a recent publication and suggested that refined alignment is needed. So the refined alignment is because the presence of insertion deletions can cause a local misalignment. So here is, I'm sorry for the people sitting in the back. So you can see that this is one example. Before refined alignment, you are identifying two variants in this region. They are very close to each other. But after you do the refined alignment, actually this is due to a lot of reads or due to a small insertion deletion there. And it's caused by the misalignment. So you can see all those two, both of these two potential variants being identified is gone. And those are really because of the alignment errors. Comparing to the initial alignment, the key difference between that is it minimizing the mismatches for one read. That is for the initial alignment. And for the refined alignment, it's trying to minimizing the mismatches across all the reads in this region. So they have pretty nice statistical model to take care of that. This is a two-step process. The first one is to identify suspicious regions, including the known insertion deletions or the regions that we have some indications of there's indels there. And the second step is to conduct the refined alignment, and which include enumerate potential haplotype candidates in that region and conduct gapless alignment on all haplotypes and calculate likelihood for each haplotype. So we went through the mathematical formulations last time a little bit. But the point is doing this sometimes it can be really, really powerful to help you to reduce the number of false positives. And we have seen, especially for the regions that you see that two variants you identified are very close to each other. Those are really suspicious regions. And we further talk about base quality recalibration. So base quality reported by all major sequencer platforms are not really accurate. So you can see this is one example from high-seq technology, Illumina technology, and the red dot here is X here is the reported quality, and Y is the empirical quality, the real quality. So we can see their reported ones are by far not as good as they are over too optimistic regarding their reporting. And this is for the machine cycles, so you can see from very beginning of the race to the end of the race, and across the board they were kind of being too optimistic, and so is for different type of dinucleotides, they were all needs to be corrected. And the way they do that is these higher than empirical quality scores provide false confidence in the base cause. And recalibration can be conducted based on multiple covariants for each individual base. This includes the reported qualities, the position inside the read, the sequencing context, mapping quality, and there's additional things that you can think of as well. Okay? So still remember that part? And we talk about variant identification, which depends on experimental designs, and so it's not that straightforward. So if people tell you, I already sequenced the DNA, identified the variants, you want to know that what is the experimental design and what tools they used. So it really depends on the experimental design. For the deployed genome balanced design, this is one example for here. So you can see this is genomic location. This A is the reference, and you have 11 reads cover this region. 10 of them are G. So you probably think, oh, this is a homozygous G here. This A probably is a sequencing error, a certain type of error. However, if this is unbalanced design, that means if it's for the cancer genome, okay, somatic mutations, and only a small proportion of the cells will have it, and so you can see this may tell you that a small proportion of the cell has the mutation and retain this as A. If this is a pooled sequencing, it depends on the total number of samples in the pool. Only a fraction of variants are different from others, especially for rare variants. So this is possibly that 1/10 of the person in this pool have this variant. And for the RNA sequencing, it also can potentially be two alleles. Their expression levels are different, and that causes this kind of imbalance. And variant identification based on sequence from multiple individuals, that's another topic we talk about. And for most of the applications, we don't really need that unless it goes to the low coverage sequences like 1,000 genome sequencing is only 4x coverage. Right? And we talk about variant quality recalibration, and this is so the idea is training on highly confident known sets to determine the probability that other sets are true. So basically, for the people for the variants that you are really confident they potentially are correct, especially for the ones that in dbSNP and 1,000 genome, and then for each individual variant you have so many covariates. And based on the gold standard you identified, you can train a machine or machine learning type of approach. And for all the other variants, and based on this machine, you will recalibrate the probability that this is right or wrong. And this is the variant quality recalibration part. And briefly touch base the structure variations. So these are different type of structure variations. As you can see what their patterns look like. And we have talked about the methods for next generation sequencing study structure variation. There's two general category of method. The first one is the methods based on tag densities. So how many reads I have for each individual location. And there's also two subcategories. which is the method using depth of the coverage to compare the different regions, and also for the case control studies to compare for the same regions. So if you go back to your notes, you'll find it. And the second category of methods are the ones that using pair-end mapping. And so basically, you know that one end is here, the other one is here, and there's a cluster-based approaches for the genome rearrangement, and the other one is for the distribution based on the distribution-based methods. So basically, that's evaluating the distance between two pairs and how that can give us an indication whether there's a large insertion deletions or other type of structure variations. Okay, so this is everything that we talked about last time. So Tatiana, it was pretty intense, right? It's a lot of information covered. All right. So, but don't worry about that. We will have homework for you to practice that. That sounds comforting? Okay. So, coming back to the lecture for today, and this is a summary slide that for the major components, all the major components in DNA sequencing analysis. So, start from the raw data, and it can be FASTQ or can be from different platforms. And then the first one, we need to do the base. The quality filtering. So we want to make sure that the rays that we are using are good rays. And then we need to further do the alignment, and then do the refined alignment. So these are the things that we really need to process for the DNA sequencing practice. And further, that we'll do the variant calling and the variant calling quality recalibration. These are important, so we already covered this part. And the next part is the variant functional prediction, which we will talk about today, and the rare allele association studies. And after all these things, that we need to go through the further validation and to make the real biological implications. And we'll talk about today is the variant prioritization, and including the variant annotation and functional predictions. And in the end, we talk about a little bit about statistical analysis of rare variants. Okay, for the prioritization part. So the needs for variant prioritization is really important. And because next generation sequencing will identify a large number of variants, okay? And most of them are passenger variants. There's no specific biological functions. And it will identify a small subset of functional important variants from large amount of sequencing data. It's important to pinpoint potential disease-causing genes and causal mutations. So this is the table that we are having from the last time. So this really tells you for different type of experimental settings, what's the number of variants that we are expecting to discover. And if this is a European whole genome sequencing, we are expecting 3.3 million variants. If this is the exon sequencing, it's about 20,000 to 40,000 variants. If it's African, and we are expecting 4.3 million variants, African for the exon sequencing, that will be 23,000 variants. And if this is the multiple samples, then this is the total number of variants we are expecting. So for the example that shows in Tatiana's class, one of the examples, so you see the 3.3 million variants. I suspect that was the whole genome sequencing, unless they did something wrong. That was my question. So, yeah. So this is really something that gave you some indication before you go into deeper whether your study had some problem or not. So if you are identifying something, the number of variants you identified is way off of this number, expected number, you may find that it's not trustworthy and there's some problems. So for the variance prioritization, so we will, for exon sequencing, we will identify anywhere from 20,000 to 40,000 variants. And how do we further do the prioritization? Of course, if we have the luxury of family information, and that is really helpful filter for us. But for a lot of studies, and those information are not really available as well. Okay, so doing the variance prioritization is really important. And there's several approaches. The first one is based on the genomic loci to do the variance prioritization, including introns, exons, junctions, untranslated regions, non-coding RNAs, promoters, enhancers, and those regions that potentially have biological information. And the second type is to do the annotation based on the population. So a DB SNP or Southern Genome or Seattle SNP, those kind of, especially the first two, you are expecting those people are generally healthy individuals. If you study a rare disease, and those are pretty powerful filter to help you to get rid of the variants that you don't need to. And so this is really the population-based variant prioritization. And the third one is the molecular function-based, including whether some of the variants will change the protein structures, and those are generally will be the non-synonymous variations, and or whether it will change the splicing patterns. That will be the whether the variant is in the splicing sites or within the alternative splice exons or synonymous variations. So we'll go through this a little bit. And we were also interested in some of the variants will be in the gene regulation level, and in the promoter that they are very likely to be the transcription factor binding sites, and in the 3' UTRs, those are microRNA binding sites. So these are the things that are not necessarily you will have everything covered for the exon sequencing, but for some other type of sequencing project like targeted sequencing, whole genome sequencing, first of all, that would be very valuable. But for some other projects, you have targeted sequencing, not necessarily exon sequencing, and this will be very important as well. Okay, so the annotation based on genomic loci and based on known gene structures, and those will be the exons, introns, and all those different structures. And there's three major sources of RefSeq, UCSC gene, and Ensemble databases. Those are three major databases you want to go through. And also, for the genomic loci part, it can be the evolution conservation. Okay, some people that suggested, I mean, it's very reasonable that conserved regions are more likely to be functionally important. And those informations are really available in the genome browser, UCSC genome browser, or other sources. And the third part is relies on other type of functional genomics data. Okay, so these are known gene structures, evolution conservation, and some other type of functional genomics data. And that includes the transcripts that are expressed in organs or tissues that's most relevant to disease. And that is one way to help you to narrow down which part of variation is important. And but that is sometimes quite difficult. And if you study alcoholism, people don't really want to give you their brain and to do the RNA sequencing to figure out which part of the brain is expressed. And sometimes you don't know which part of the brain you are looking for, so it becomes a problem as well. So, but it can be quite difficult, and sometimes it can be misleading. But there's another piece of information that most people are not really looking into. There's a lot of data in the public domain, especially the ENCODE project generated a lot of annotations for the human genome. For example, the promoter regions, and most importantly, they identified a lot of enhancer regions. For the promoter regions, it's kind of simple, right? We just get a transcription starting site, go upstream a few thousand base pair. We're thinking that the promoter region are there. But for the enhancers, it can be randomly anywhere in the genome. But ENCODE project really help us to map those regions out. That's important. And also the DNA hypersensitivity regions, and from molecular biologists, that you know that there are most likely there's a lot of transcription factors or other DNA binding proteins are working there and to make biological functions. So based on genomic loci, this is the known gene structures, which is straightforward, and evolution conservation, this really makes sense, and some of other type of annotation that derived from functional genomics data are important. And most of this information are actually just one of the tables in the UCSC genome browser. If you can do reasonably programming, and those regions can be your variations can be mapped to those regions. Okay? And so that is the annotation based on the genomic loci. And the second general category is the annotation based on the populations. So we will talk about the DB SNP Actually, you can see that this figure tells you the data flow in dbSNP, and this is sublimation process, and the data will be hosted in NCBI, and the retrieval and the applications. And actually, the dbSNP information was submitted by different laboratories, including research labs, sequencing centers, or databases, and they all have a SS number initially, and those are the submission numbers. And then they were stored in the NCBI, but for each genomic loci, hypothetically, we are hoping that there will be one RS number assigned to them so that we can later refer to those. So they were hosted by NCBI. It includes actually, it's called dbSNP, but they have the information more than SNPs. They have insertion deletions, microsatellites, and there's a lot of other type of genetic variations. And the records include more than human. They have actually so far 55 different organisms. That's a lot of information to go through. So if we study animals, and this is a good source to look into as well. And now it includes 1,000 genome sequencing data and the NHLBI exome sequencing data. So they have multiple data sources in their database already. And this is one stop that you can really go to, and because it also included 1,000 genome sequencing data. But sometimes you got to be careful using it because 1,000 genome sequencing have many different subcategories and people from different area in the world, and dbSNP not necessarily distinguish that. So sometimes you have to go back to the 1,000 genome sequencing database and to look into it. And the current version, and actually they just updated a couple weeks ago, and 2-3 weeks ago, and it's 134 version and have a lot of information. And the next annotation based on population will be the 1,000 genome sequencing project. And Tatiana also talked about a little bit. And they include the 5 major populations, including the European, East Asia, West Africa, Americas, and Southeast Asian ancestries. And the phase 1 study, they have almost 1,200 individuals sequenced, and we know those are pretty low coverage 4X sequencing, but it gives you the very good estimation on the allele frequencies of these individual populations. And they are planning for 500 individuals for each population. So there will be 2,500 people will be sequenced in the near future. And another one will be the Seattle SNPs, and this is only for the inflammatory responses in humans, and so that is a very important source as well. And next one is HGMD, that will be the human gene mutation databases. And those are not really help you to rule out anything. Those are just help you to identify whether your variation has already been identified or not. And those are really humanly curated database, and a lot of people are using HGMD as their gold standard when they do research. Okay? So we talk about annotations based on the genomic loci and annotations based on the population. Now becomes the annotation based on the molecular functions. This is the third general category. The first 2 categories more like you are query existing database. But for this category, there will be a lot of predictions and bioinformatics efforts involved. And just to go through a little bit of the variations and different type of variations, the first category is the nonsense variation, which is within an exon and translated amino acid change to a stop codon. This is really causing a huge problem. So if you are thinking that there's one gene, it will transcribed and then translated, if there's one variation that causes one amino acid sequence substitution, and that is generally okay. But if it's become a stop codon, you are running into a big problem because a big chunk of the gene has already been doesn't exist in your final product, protein product anymore. And the second type is a frame shift. It's within an exon and translated, and insertion or deletion interrupts the reading frame. And this is a big problem as well, because we know that for the codon, it's every 3 nucleotides become amino acid. But if you have one more inserted in that, that totally, shifted the rest of the amino acid sequences, and that causes a huge problem. And the next one is the coding sequence non-synonymous variations. This is the nucleotide difference that will change amino acid sequence substitutions, and that sometimes can cause a problem. And splice 5 or splice 3, we'll go through this a little bit. This is very important. It's in the first 2 base or last 2 base of the intron. And these regions generally are where the spliceosome sits on. And sometimes if that part has a variation and it's not regulation, the spliceosome cannot form there, so you cannot process the splicing. That's why that for most of the major predictions, and the splicing 5 and splicing 3 are quite important. And coding variants, synonymous variants, is within an exon and translated, no protein amino acid change. And a lot of people consider this type not very important, but I will tell you there's something that new development found a lot of synonymous variations are actually quite important as well. And next category is the variations occurred in the 3' or 5' untranslated regions. And these are the regions are within the exon but not translated. So Tatiana made the comments that the exon sequencing, most of the exon sequencing, the capture part doesn't include the 5' UTR and the 3' UTR. That is correct, and that's because when they designed those chips, those regions are not included. But actually, those are also considered part of the exon. So the terminology becomes a little confusing, but they are just not on the chips. And they are sometimes they are not translated into the protein product, but they are doing a lot of translational regulation, or at least post-transcriptional regulation. And next category is the regions that near gene 5' or 3' end, and the people consider there's a lot of regulatory components to control the gene expression are located in those regions, and also the intronic regions. So these are the terminologies that we will be using, but I'm sure that most of you are familiar with this, given how many molecular biology classes you have already taken. Okay, so let's go to the most important type of variants, which is the non-synonymous variations. The importance of this is it causes amino acid substitutions and thus affecting phenotypes of a host organism. All right? It constitutes more than 50% of the mutations known to be involved in human disease. And there's evidence also shown in OMM database and HGMDs. The human population have anywhere from 67,000 to 200,000 common non-synonymous variations, SNPs. Each person has 24,000 to 40,000 heterozygous non-synonymous SNPs. Okay, and this is I don't know how they get this number, but this is based on one of the references, and they were talking about this. And this is important point, is non-synonymous SNPs have a lower minor allele frequency distribution than those that are predicted to be functionally neutral. Okay, so this is a very important point, because this tells us that the importance of these non-synonymous variations is the nature selection actually selects against of this type of variations. So once those occurred, a lot of those individuals will not go on for the next generation. So they were eliminated from the new general population. That's why they have a lower minor allele frequency distribution. And then those are predicted to be functionally normal, neutral. The general strategies for non-synonymous SNPs functional prediction, and actually disease-causing mutations are more likely to occur at positions that are conserved through evolution as compared with positions that are not conserved. So this is a boring sentence, but it's so important because this is the base for most of the prediction algorithm. So let's go through this again. Disease-causing mutations are more likely to occur at positions that are conserved throughout evolution as compared with the positions that are not conserved. And this is, so you can see from the handout, from the slides. And for the first figure here, the X here is the conservation. So this is the lower conserved regions and the higher conserved regions. And the red actually is a disease-causing mutations. So you can see with the conservation increase and the probability that relative mutation probability goes up. So it becomes more and more important. And for the second one here is X here is actually the structure. So the solvent accessibility of a mutation site is related to the structures. And towards this end, it becomes more and more structured and less structured. So you can see that more and more structured regions, and they are more likely to be related to disease-causing mutations. Okay? That's why that for most of the algorithms to predict the protein functions and variation functions on the protein structures are based on either structure, either sequence, structure, and or annotation. Okay, and so the general workflow here is for the input protein sequence and amino acid substitutions. So it goes through the structure pathway, and for the structures, and you can see that search a protein structure database, and then model input sequence on the structure of the top hit, and then further calculate the probability. The second part is get a sequence and then search a protein sequence database and obtain homologous sequences. So basically do the comparative genomic analysis and then only find out the regions that are evolutionary conserved. They are more likely to be important. And the third one is the annotation. So far there's more than 24 of algorithms predicted just to prioritize non-synonymous SNPs. And there's a very good review, and I encourage you to go there. Actually, the author of this is the developer of the SIFT. Okay, we'll go through a little bit in a couple of slides. And I think Sean Mooney invited her to come here give a talk a couple of years ago. So the most famous algorithm most people are using is the SIFT. It's based on the sequence homology. It doesn't based on structure at all. And the second one is the polyphon. It's based on both the sequence and the structures. Okay? So which algorithm to use? And of course, we always want to pick the best performing algorithm. But the direct comparison among methods are really difficult because each individual method, they were based on different assumptions. Okay? And the performance is dependent on the training and the test data sets. So if you read those papers, every algorithm will tell you they outperform others. Okay? It really depends on what type of training data sets they are using, and sometimes those results are not really trustworthy. But direct comparison between different methods are really, really difficult. And the problems of the structure-based variation, you may think that, so we talked about the SIFT and the polyphon. SIFT is only based on the sequence homology, and the polyphon is based on both sequencing and the structure. So you may think a polyphon is using more information, it must be more accurate. But sometimes it's not necessarily true. For the method that based on structure-based method it's the last prediction. The reason is we have very limited amount of structures available in the database. So your prediction sometimes it's just not included. And it can be misleading as well because structures are predicted as silos, not in the contents of cellular environments. We know that when the people sitting in the lab to do the protein structure identification, they really do those in the tubes, and it's really outside of the cellular environment. Sometimes those misinformation can be really misleading. For the sequence-based algorithm, the homology and the orthologs in different species can have different functions, but this will be violating their assumptions. So sometimes because of this sequence-based study, algorithm is not really accurate as well. Okay, and the last point here is the prediction is really for the guide for the experiment, not for the clinical settings. That is what most algorithms they will tell you. So this is for hypothesis generation. You are welcome to use this to design further experiment, but don't use this prediction in the clinical settings. And just because of the low accuracy for each individual methods. So it still comes to the question that which algorithm to use. And it's really, really hard to say. Different algorithms have different assumptions. But probably because of this, you want to try different things. Maybe that some algorithm is good for one of the assumptions and others is for another type of assumptions. So you can use the multiple algorithms to predict things, and they may each pick up different things. So which is really fine. Okay. And non-synonymous SNPs prediction for complex disease. This really works well in Mendelian diseases, but how about the complex disease such as hypertension, diabetes, or heart disease? We really don't know about that. The reason that it's good for the Mendelian disease is most of these algorithms are based on the assumption that evolutionarily conserved regions are less likely to be problematic. And sometimes for the complex disease, this may or may not hold. For the Mendelian disease, I'm sorry, yeah. And complex genetic diseases are those that cannot be mapped to single loci, which might indicate an interaction between multiple loci or an interaction with the environment. And so there's two different models of this. The first one is the common disease, common variant model. And the second one is the common disease rare variant model, right? We're talking about this. And for the GWAS, that most people are based on this common disease, common variant model. So diseases are caused by interactions of multiple genes with less but the common variants. But for common disease rare variants model is diseases are caused by many low frequency variants. And Mendelian, some people consider these are the Mendelian disease, but by different variations. Okay, whether it's a right or wrong, we don't know, but some people consider in this way. And this figure really tells you that whether some of the prediction algorithm can potentially fail. So this is a paper published in PNAS. So X is some kind of a biophysics evaluation, and sometimes they don't worry about that. But you can see there's for different lines, and the first line is actually the red one is a Mendelian disease, and the yellow part is a neutral model, and then the normal variations, which is variations in dbSNP, is green, and these the blue dots are the complex disease. So if you go to the left side, the more the left side is, and the better prediction power of these algorithms are. But you can see that for the complex disease, some people evaluation is this really doesn't do very well. Okay, so you want to use that as a precaution. Are we okay with the non-synonymous variations? So basically, there's a lot of words in these slides, but there's some key points that I want you to understand. The most of these non-synonymous variation predictions and are based on the one important assumption that evolutionarily conserved regions shouldn't be messed up. Okay, if it is, you will get into trouble. Okay, so that is their basic assumption. There's two types of information that people use. The first one is the sequence along, amino acid sequence along. The second one is the sequence plus structure. And so each different algorithm can map to that different categories. And the third information I want to deliver here is these predictions, whether they are working for the complex disease at this stage, we don't know. I think those things still need to be tested. Okay? Can we move on to the next one? And for the synonymous variations, these are traditionally called the silent mutations, and they are the ones the nucleotide variation that will not cause amino acid sequence changes. So most people just ignore this part. So if you can see that a couple of examples that Tatiana presented, they were a couple of filters based on the Southern genome filter, and then they only focus on non-synonymous variations or splicing variations, not on the synonymous variations. Okay, and they are traditionally called silent, and but they may change. They may cause changes in the protein expression, conformation, and functions. And this table actually listed all the current, most of the current known that synonymous variations that cause different disease. So you can see this vision, muscle, kidney, and each individual tissue that with human disease, they have potentially a lot of synonymous variations has been discovered in the previous studies that they can cause disease. So we really shouldn't ignore this part of information. Okay. And synonymous, a recent study published in 2010 reported that synonymous SNPs share similar likelihood and effect size for disease associations between disease and SNPs curated from 2100 reports. So they are just as important as non-synonymous SNPs. Okay, so we really shouldn't ignore those. There's a multiple molecular mechanism for the synonymous SNPs, why they are important, why they can cause disease. The first one is the splicing regulation. Okay, so if you can see that this is 3 different exons and this introns, we know that this part needs to be spliced out, and eventually the final protein product include 3 different exons. If this one exon is not included in the final product, and this is cause major differences. And you can see that there's these splicing regulation are regulated by a lot of RNA binding proteins, and one of the proteins is the so-called SR protein, and we are plotting here. But if you can see that SR protein, if it can bind there, that will promote this type of final product. But if there's a variation, genetic variation in this region, although it doesn't change the amino acid sequence, but that will potentially affect whether this protein binds here or not. And if it's not binding here, and you will get into this type of protein product. And just one nucleotide difference. So if it's a non-synonymous variation, most of them will change one amino acid sequence. But this one synonymous variation will cause the entire protein domain be lost. So this sometimes can cause bigger physiological effects. Okay. And also that the second molecular mechanism is the global stability and local stability issue. Okay. So this is you can see that this is the RNA, the messenger RNA, and we know that messenger RNA really forms this type of secondary structure. And the only difference between these 2 structures are this is a double-stranded RNA formation here, but here is a hairpin loop. There's a budge here. Okay. The reason for these 2 type of things is there may be one genetic variation in that region, so that they form this way rather than this way. Okay. And it's reported that this type of structures are much less stable than this part. So because of that one variance, even though that one variance didn't change amino acid sequences, but that will really change the global stability of different messenger RNAs. And we may end up with more RNA protein product of this form rather than this form. Okay, that is really a very subtle but important regulation. Sometimes it happens in this way. If this variation happens very close to the 5' end of the gene, okay, like this, in the 3' UTR region, and this forms this kind of a bubble. For this form, we know that a lot of variations, a lot of proteins that start to do the translation initiation will happen in this part, in this region. So if this is a more stable region, and that the translation will become harder to initiate. But this part, the translation will become much more easier to be initiated. And the end result for that is we will have less protein product on this part because it's hard to initiate translation, but more protein product in this form, just because of the one variation in the 5' UTR region. Okay? So you can see that the global stability and local stability can both affect it. And the third part is the genome trend in the codon usage. I think it was 3 years ago, there was a science paper published that they say there's some variation. They don't change amino acid sequences, okay, but just because they use different tRNA and the availability for different tRNA are different, so that can cause the translation become faster or slower. Okay, the faster and slower sometimes will change the protein conformation and eventually lead to different protein products. And that's just a challenge the traditional view of non-synonymous variation that don't change amino acid, doesn't matter. But this really does matter. And the last one I want to go through here is the ribosome pulsing size and the co-translational folding. So you can go to the regional paper. We'll put that into the course website. But the point here is when the translation is going, and sometimes that there's some site will stop, cause the ribosome to pause there, and therefore doesn't further do the moving. So even though their RNA sequences are almost identical, if they become a protein sequence, it will be also the same. However, their rate of translation will become different. That will still change the protein conformation and therefore cause some problems. Okay? So these are some of the mechanisms that just challenge the traditional view that why only non-synonymous variation are the focus. And the synonymous variation are not always the silencing mutations, and sometimes they are important. Okay? In general, that synonymous variation, the molecular mechanism, have 3 different categories. The first one is it changes the way that post-transcriptional regulation occurs. This includes the splice site, and the splice regulation, and the microRNA regulation, if the variation happens in the 3' UTR of the region. And the translational control, this includes translation initiation, early translational elongation, late translational elongation, and co-translational folding. And sometimes it changes the protein stability as well. Okay? So even though they don't change amino acid sequences, they can have pretty big effect on the cells that we're working on. So now let's go through the methods to study functional predictions on synonymous variations. The first one is the splicing sites, and this is actually included in most of the prediction algorithms. And so this is the splicing site. Don't confuse with those SR protein binding things. We'll go through that later. But this is some site that is very close to the junction and axon, intron and axon boundaries. So it's maybe just 2 base pair away from the junction. And those are the regions where the spliceosome form there and then to conduct the splicing process. If those are messed up, and this axon will not be accurately spliced. And those really can cause a very severe effect. But the second type of splicing is the splicing regulators. So what happens is, if you see this is the axon, this is intron region, and some of the variations in these regions will change the RNA binding protein binds in those regions. And there's many different types of such regulators, including the axonic splicing enhancer. That means if that enhancer is that variation is in this region, and that the protein will bind in the enhancer region and further promote the splicing. So sometimes it has exonic splicing silencers, or intronic splicing enhancers, or intronic splicing silencers. So there's multiple forms. And there's many databases so far available to document where these different splicing sites are, and including the human splicing finder and ESE finder, rescue ESE, and all those things. So for general practice, if you identify a variant in those regions, you want to overlapping with these regions to see whether they are locating into a region that can change the splicing regulation. Okay? And our approach actually is a little different because just because that variation locates in the region that can have a protein bind doesn't mean that variation is changing the binding itself. Okay, so our way is we generate two distributions. One is a distribution for the non-binding event, and the other one is the distribution for the binding events. Okay, so we know that for the binding events, the score distribution look like this. For the non-binding events, the score distribution look like that. And then we look for calculate the score for minor allele and the major alleles, the variation before and after the variation, what the scores are, and then trying to have an inference that whether that variation can cause you change from one population to another population. So it's not just overlapping it, it's more quantitative evaluation. We think it makes a lot of sense. And we did apply this on the 1,000 genome data. 1,000 genome sequencing, they publish is exon sequencing data, and there's a couple of thousand exon variations. And when we look at this, you can see this non-synonymous variations, and Y here is a minor allele frequency. So for the non-synonymous variations here, and the minor allele frequency is this much. Okay? For the synonymous variation, the last one, for the synonymous variation, the minor allele frequency is this much. It's higher than the non-synonymous variation. Remember the 2004 paper in the PNAS talk about that in general, disease-causing variations have lower minor allele frequency, right? Just in a couple of slides before this. So this is, from our data, this really proved that. And this is from 1,000 genome data, from our analysis. And this bar here actually is also the synonymous variation, but it also locates into the exons that can be alternative spliced. Okay? So if there's a synonymous variation in the exons that can be alternative spliced, it didn't change the allele frequency much. And this one is the synonymous variation, okay? And also they are changing the RNA binding protein binding patterns. So we can see it didn't change much as well. However, once we have the synonymous variation and also it locates in the alternative spliced exons and also change the RNA binding protein binding, if all these three components meet there, and the minor allele frequency becomes as low as non-synonymous variations. This really suggested some of these variations that can change the RNA binding protein binding and therefore affecting splicing. And once these two conditions are meet together, and you want to put a little bit more strength into higher prioritization for those kind of variations, okay? Even though they are synonymous variations. Okay? Actually, we have the code to conduct this type of work. And this is just published, and we just got an acceptance letter maybe a week ago, and they said it will be in the PubMed very soon. So another type of variation. So next I'm going to talk about the variations in the promoter regions and the 3' UTRs. Okay, and in the 3' UTRs, it becomes really easy to understand. We know those are enriched of microRNA binding sites and RNA binding proteins in the 3' UTRs. And they usually, so hypothetically, if there's a variation occurred in those regions, that can change these kind of regulations. And they are usually on the seed sites and the position 1 to 8 of the microRNA target sites. Okay? And so it's become very simple. You just get the variation, you know where the variations is, and you're overlapping that with the microRNA binding sites, and you can see whether it's in the microRNA binding sites or not. If it's in the seed site, you probably want to consider they are higher priority. They may have a little more biological function. Next is in the promoters. They are usually considered as where transcription factor binds. So the general notion is if there's a region or there's a transcription factor binds in the promoter region upstream of the gene, if there's a variation here and transcription factor doesn't like this region anymore, therefore that will affect the gene expression levels. Okay? And the most methods doing this promoter SNP prioritization is identify the SNPs residing in all the known or predicted transcription factor binding sites. So you know these are the regions that transcription factor binds, and you see whether I find a variation in those regions. If I did, probably it's important. Okay? And these are the methods that are available just for that type of approach. And this is the work that just published actually from Sean Mooney's lab, and he was here in Indiana, right? And I just made this lecture and dig his publications, and they are having a new approach that using more features, not only whether they are in the transcription factor binding sites or not. They use more features such as distance to transcription starting site, GC contents, flanking nucleotide sequence, and many more, and then using machine learning approaches to do the training, and then to see whether they can find prioritize the variations in the promoter regions. And actually, they use the supporting vector machines. Okay? And the limitations of all these approaches are not really considering the degeneracy information for the transcription factor binding sites. So you consider this is a transcription factor binding site. Have you ever seen this type of logo? Most of you did? Okay. So what that means is this is a 8 base pair motif that transcription factor like this 8 base pair. Okay. However, they really prefer this part to be a G. Okay. And this part prefer to be a A. This part can be a or G, and if it's A or G, both are okay. And sometimes this type of degenerate information are built in the transcription factor binding sites, and most of these motif finding tools are not realizing that. They're just overlapping. This is transcription factor binding sites. This is a variation, and if it's in, that means this variation is important. Sometimes if the variation occurred here, you're in trouble because that will completely knock out whether this transcription factor can bind in this region or not. However, if the variation occurred here, it's less important because the transcription factor really doesn't care about that base. Okay? So those information needs to be considered. So we had this approach, which is very similar to the RNA binding protein starring. And this is a different score calculation, and you don't have to get into it. But the point here is for the known binding sites, we have a binding sites distribution, and we have non-binding sites distribution. And then you calculate the variation in the reference allele or alternative allele. And then after you get in here, you want to see whether this one variation will change the binding score big enough to shift from one distribution to another distribution. Sometimes it can be a gain of a function. Sometimes it can be a loss of a function. So this type of approach, sometimes you really get the gain because of that variation comparing to the reference genome. It gained the transcription factor binding. And that sometimes if it's a cancer-related gene, you may get into trouble for that. And our results on one of the bone studies actually identified the 3 transcription factors and their binding sites. And this, I think, so these are the arrow part are the regions that are the sites that are predicted to be important. Actually, the third patent was biologically validated using luciferase assay. So the point I'm delivering here is still challenging the traditional notion of only non-synonymous variations are important. there is a lot of synonymous variations are also functionally important. Sometimes they are even more important. And because they are not changing one amino acid, rather they are changing, you have more or less of such protein, or causing an entire protein domain missing. And those are really, really important. You need to think through. And on top of that, for the non-synonymous variations, that most of the current prediction algorithm is based on the evolution assumption, and you it may or may not fit into the complex disease scenario. So just think through that. And so the point of what I'm saying here is not really teaching you something new. Rather, so when you read papers, you see they are considering this is a different level of filters, identified 40,000 variations, 38,000 of them are in southern genome. Let's get rid of them. That's fine. And you still have 2,000 left. And that 2,000 of them have 1,000 in the exon regions, and 50 of them are non-synonymous variations. Let's only focus on these 50 non-synonymous variations. And sometimes you are losing a lot of information. That is one of the reasons probably many of the disease people didn't find the gene that are there. And there's many different tools for the variation annotation. And in the earlier, actually this is a trivial problem, because most of people, if you can do the programming, you can really do it. It's not difficult, right? But because of this, become more and more important problem to solve. So there's algorithm, a tool developed called an ANOVAR. It's an annotation for the variations. So basically, it's a functional annotation of genetic variants from high-throughput sequencing data, and published last year in NAR. And this is most of the people are using this tool right now to prioritize their variations. So they have the gene-based annotation, region-based annotation, and filter-based annotation. The filter-based annotation is just something that you can say, get rid of all the reads that are in Southern Genome Project, but only the ones that the man-allele frequency is larger than 10%. So you can do more complicated things than using this tool without needing to do the programming. And the Plink-seq, and there's another tool that you can do a little bit annotation, but I think all are covered by the ANOVARs. And many popular tools have their own wrappers, like the SIFT, Polyfin, and these are the things that you may want to go back to their own website to download the code to do the annotation. And sometimes you also need to customize the approach. You need to write your own code based on the existing database to do that. Especially some of the databases are not standardized database, and you probably want to write something to help you to prioritize your variants. Okay? So is that clear? So basically, this entire part is tell you that non-synonymous variation and synonymous variation are both important. So, but put a little bit consideration before you're using it and after you get the results. Okay? And very quickly, go through a little bit statistical analysis on the rare variants. I'm not going to give you any equations. And if you are not satisfied, there's plenty of paper gives a lot of equations that doesn't make any sense to me. All right, pages of equations. For this part, and so we know that for the genome-wide association studies, there's a lot of very established algorithm to deal with that. But here we are talking about the rare variants, which is a new feature for the next generation sequencing data. The rationale for this part is multiple rare variants, both within the same gene and across different genes, collectively influence the expression and the prevalence of traits and disease in human populations. Not one gene or not one gene. So it's multiple genes, and they're collectively influence the phenotype. So that's the basic rationale. There's many different scenarios on the DNA sequencing variation, distinguish cases and controls. Okay, controls are the general population. Cases are probably the population that the group of people that are sick. So for this scenario, you can see. I think this battery is dead. Okay, for this scenario, you can see this is very simple single locus common allele scenario. And this is the case and control, and there's more in case and less in control. Simple, right? And this is another type of scenario, which is multiple variants collective frequency. So you can see that it's not one or two variants in both case and control, but in the case, there's a much more number of variants in that region. And sometimes this collectively will be a good association to a certain disease. And the third scenario is multiple variation synergistic disease effects. For example, you can see that for the case, in the case part, this variation, some of the variations, they are always occurring together and they have follow certain specific patterns. But in the control case, such the patterns, clear patterns are not there. So there's a possibility that multiple variants have this synergistic effects. And the fourth scenario here is multiple variants with specific regions. So you can see that if you look at this entire region in the whole, you probably don't see too much difference between case and control. However, most of the case, the variation occurred in these two regions, but in these two regions, in control scenario, there's nothing there. And so this is a next scenario that may have this the effect for these rare variants. Okay? To do the statistical testing after you identify so many variants, the first one is a single marker test. So basically, it's very simple. It's just to test each variant individually with the use of univariate test and assess the significance of test after doing the correction for the multiple hypothesis testing. Okay. So this is, in general, the short conclusion for that is it won't work. Okay, just because the power is too low. We have so many variants to correct the multiple hypothesis testing, so we are not going to get the good power for that. So this is one of the simulation studies in this Nature Genetics, Review Genetics. So you can see that if the red one, this is a kind of common variations, okay, and you probably, if the allele risk is about 5 in this region, and you need about how many 1,000 samples to identify to make it significant. However, if this allele frequency is 0.001, which is a rare variant, and with a relative risk of 5, so you can see that you need over how many 10,000 samples, 20,000 samples to really identify one small effect. And actually this is not a very small effect. This relative risk is already 5. So for this single marker test, this will not work. And the second type of test is a multiple marker test. So you can see that this is a type of association studies, very simple regression type of approach. So you instead of using one individual SNP markers in the equation, you use multiple collectively. Okay, and sometimes people think, oh, that reduces the number of tests, hypothesis testing you want to correct, so you have more power. But keep in mind that it does have a higher power than single marker test, but methods include Fisher test and this test are multiple regressions, and they require, most of these methods, they require multiple degrees of freedom. And if you're a statistician, you know that will lower the power as well. So multi-marker tests sometimes have issues. And it's sensitive to allele frequencies and can reduce the power with too many variants as well. And the real method that people are using currently is something called collapsing methods. So the rationale for that is given that single marker test involves correcting for multiple testing, and that multi-marker tests have a large number of degree of freedom. And another approach is to collapsing the genotype. Single marker test, too many tests to do the correction, and multiple marker test is too many degree of freedom. Both reduce the power. And for the collapsing method is defining an indicator variable x for each individual. If rare variant is present in that individual, and you put it as x is equal to 1, otherwise it's equal to 0. So this is the general workflow of this collapsing methods, and whether you agree this type of assumption or not, that's a different question. So the problem here is if the frequency of rare variance is large enough, or if there are many of them, then the percentage of individuals having this type of variations could reach 100%. So you really don't get anything out of it. So there's a lot of different variations on the collapsing methods, and this is generally these are the methods that fell into the category of method based on the summary statistics. Okay. And there's a method based on similarities among individual sequences, and there's a method on multiple regression and data mining methods. So I'm just listing there, and I don't require you to know too much about this area, but what is my hope that once you get to that stage, you get the data and you know where to look for the help. There's a big research area about rare variant association studies, which can be pretty difficult. And there's a very good review published in Annual Review Genetics last year, and feel free to go through that. And the summary for this part is many methods, but evaluation is really tricky. Okay. Again, so this, when we go back to the previous slide, there's so many different methods. So I just feel it's amazing. I was in a meeting earlier this week, and it was the statistical methods in next generation sequencing data analysis. And at the time that not many people have data, actually most people don't have the data yet. And there's a lot already, probably 24 methods already developed. And people start comparing those and to make us say this is a generalized form and that's a special case. I just feel it's really, really amazing that people can develop those statistical models without any real data. And so that comes to the problem of that the evaluation becomes very tricky, because even though we have the real data, we wouldn't know that what is the right answer for that. But without real data, and people do a lot of simulation, and of course their method, the way that they do the simulation will follow the assumption that their method is based on, so their method always performs better than everybody else. And that makes the evaluation becomes really, really tricky. And the power and the robustness of these models needs to be assessed in a wide variety of contexts, so we don't really know too much the performance of different approaches. And finding that a set of rare variants seem to be collectively associated with phenotype in no way suggests that these are the functional or causal variants. They are just associating with the disease. They are not really necessarily to be the causal ones. And if you want to identify the causal ones, further experiment is needed. And the collapsing method can be used either before or after the functional predictions. So we talk about the functional predictions and the association studies and collapsing of variants. So you can collapsing all the variants in that gene, or you can collapsing the variants that predicted to be functional on those genes. And that will be the non-synonymous variations, the synonymous variations, and the topics we talked about in the previous slides. Again, coming back to these major components in DNA sequencing analysis. So we got raw data, we need to do quality filtering, alignment, refined alignment, and the variation quality recalibration, and functional prediction, and rare allele association. And from Tatiana's lecture, and we also talked about in the first DNA sequencing lecture, that if you have additional information, and like family structures, and that is really, really helpful. Okay. And for the next 3 lectures, we are going to talk about the RNA sequencing experiment. And for the first one, which is next week, we'll talk about the biological theories on RNA sequencing experiments and experimental considerations. And we'll summarize some of the things that we cannot do without RNA sequencing, just based on array-based approach. And we also will talk about alignment here, because RNA sequencing, the alignment has become more tricky than regular alignment. There's some special strategies for that. And we will summarize the gene expression analysis. And how can we say how many transcripts are transcribed just based on the next generation sequencing data. And the second lecture, we will first have a half a lecture, and it's Milan Rotevich, and who was a graduate student in genetic department, just become a faculty member in Department of Surgery, and he has a very interesting RNA sequencing data experience, and he will come here to give a guest lecture. And then we will spend the rest of time talking about alternative splicing and the potential allele-specific expression. Okay, both of them are very, very interesting. And the third lecture, actually it should be RNA-seq3, and we'll talk about the non-coding RNA analysis and pair-end and mate-pair analysis on the RNA sequencing data, how that will help us, especially to identify gene fusions. Okay, and we'll use about half of the lecture to talk about some of the topics in the small RNA sequencing, like microRNA sequencing data projects. Okay, and mark your calendar, Tuesday, November 1st. Okay, and Aaron Torrent will come here to give a seminar series, and for 4 hours. Okay, and they have a couple of, I think there will be a couple of sales talks, but they will bring in their national presenters. So they go to every single big meetings and to say how good their technologies are and what's the science that they have done using that. And it will be very interesting. And more importantly, they will provide lunch, and that is a good incentive. Yeah, that will be in the R3 building. And actually, Indianapolis is one of the four locations they selected this year to give this seminar series. So we really want to take advantage of this. So I don't have the flyer yet. I just helped them to arrange the room today. So just the location is fixed today. So once the flyers is ready, I'm going to send out and presumably send to Lily as well, and maybe some other industries in the town, and hopefully we all enjoy that event. All right, less exciting, the second assignment. All right, I think we can only for the students who register class day. All right, so we will talk about take a small break, 2 minutes, and just sit there, and everybody feel free to go.